Hello friends, how are you? I am back with another video. Today we'll start with metabolism of phenylalanine and tyrosine. There are three major questions that are supposed to be asked in this metabolism. First is the metabolism proper of phenylalanine and tyrosine. The second is the products of tyrosine and phenylalanine metabolism, right? And the third is inborn errors of metabolism. Now we'll start with structure of phenylalanine. Since the name is phenyl plus alanine, right? Phenyl means phenyl ring is there, right? Plus alanine, and we will finally get phenylalanine, right? So let's draw the structure. First, we have to draw phenyl ring. This is phenyl ring, and this is the structure of aromatic alanine, right? So this is phenyl ring, and this is alanine. So I will write. Now I will make the ring proper. So this is phenyl ring, and now I'm attaching alanine. So this is the structure of phenylalanine, right? So now coming on to the structure of tyrosine, right? So tyrosine is para hydroxy phenylalanine, right? So this is the structure of phenylalanine, and tyrosine is para hydroxy phenylalanine. So in order to convert this phenylalanine to tyrosine, we have to attach hydroxy position at para position. So when I will attach hydroxy position here at para position, so it is now tyrosine. Now we will start with the first step, that is conversion of phenylalanine to tyrosine. So phenyl ring is there, and this is alanine. Both are attached. It is known as phenylalanine. And tyrosine contains para. The name of tyrosine is para hydroxy phenylalanine. So when we introduce hydroxy position in phenylalanine at para position, it is converted to tyrosine. The enzyme required is phenylalanine hydroxylase, and oxygen is required and is converted to water. And two molecules are required. One is incorporated at para position, and other is converted to molecule of water. This reaction is tetrahydrobiopterin dependent, and this converted to dihydrobiopterin, right? And it is again recycled by NADPH dependent reaction. If there is blockage in this pathway of conversion of phenylalanine to tyrosine, and there is absence of this enzyme phenylalanine hydroxylase, then this irreversible reaction will not take place and this pathway is blocked. The disease will be phenylketonuria, right? Now we'll move towards next steps from tyrosine onwards. Now we have converted phenylalanine to tyrosine. Now we will convert tyrosine to para-hydroxy phenyl pyruvate. So I will draw structure of tyrosine first. So tyrosine is para-hydroxy phenylalanine, right? Right. So this is tyrosine para-hydroxy. Para position we have hydroxy group. Phenyl ring is there. Alanine para-hydroxy phenyl. This is tyrosine. Now I will convert it into our second product, that is para-hydroxy phenyl pyruvate. So para is there, hydroxy position. So this is para-hydroxy phenyl, and pyruvate is there. Right. So this is converted by enzyme tyrosine transaminase. Right. Tyrosine transaminase. This reaction is transamination reaction. Right. So alpha ketoglutarate is converted to glutamate, and the second enzyme is tyrosine transaminase. The disorder that will come is, if there is blockage, this is known as tyrosinemia type 2, right? So you can remember by second enzyme is there, so disorder is second, tyrosinemia type 2, right? So now we have converted tyrosine to para-hydroxy phenyl pyruvate, right? Right. Now we will move forward. Para-hydroxy phenyl pyruvate is now converted to homogenticate, right? So how the reaction requires ascorbate, right? Vitamin C, and this enzyme para-hydroxyphenyl pyruvate dioxinase is copper dependent enzyme, right? So now we'll have draw structure of homogenticate. So this is homogenticate, right? Right. So we started from phenylalanine. Phenylalanine was there and alanine was there. It was phenylalanine. It was converted to tyrosine. Tyrosine was para-hydroxyphenylalanine. Para-hydroxyphenyl and alanine, right? So this was tyrosine. It was phenylalanine was converted to tyrosine by para-phenylalanine hydroxylase, right? Now tyrosine is supposed to be converted to para-hydroxyphenyl pyruvate. This reaction is a transamination reaction, and it is conversion of alpha ketoglutarate to glutamate. And the disorder here is second enzyme is there, so disorder is tyrosinemia type 2. If there is blockage here, right? Now the third product is para-hydroxyphenyl pyruvate. So this is para position hydroxy group, phenyl is there, and this is pyruvate. Tyrosine is now converted to para-hydroxyphenyl pyruvate, right? The enzyme is now we have third product, that is para-hydroxyphenyl pyruvate. Now it is converted to fourth product, that is homogenticate, right? The enzyme is para-hydroxyphenyl pyruvate dioxygenase, right? So it requires copper and ascorbate, right? Now para-hydroxyphenyl pyruvate is converted to homogenticate. Now I will draw structure of homogenticate. Right? This is homogenticate, and now homogenticate is further converted to 4-metyl acetate, right? So the enzyme is here is homogenticate oxidase, right? It was converted to fourth product, that is homogenticate, right? So we introduce two hydroxy groups, CH2COO. This is homogenticate, and it is converted to fifth product, that is 4-metyl acetoacetate, right? This is carried by enzyme homogenticate oxidase, and if there is blockage in this step, the disorder is neonatal tyrosinemia, right? And if so, this is the fourth product homogenticate, and fourth to fifth product is blocked this reaction, then the disorder is alkapton urea, right? Now we have fifth product, that is 4-metyl acetoacetate. Now we'll further convert into next steps. So we have now 4-metyl acetoacetate, right? It is converted to 4-fumaryl acetoacetate, right? So these are isomers. The enzyme is isomerase, right? And if there is blockage in this reaction, right? The disorder is tyrosinemia type 1, right? So now 4-fumarate acetoacetate is further converted to there and it undergoes hydrolysis, right? Right? So it is converted to two products, that is fumarate and acetoacetate, right? So fumarate can enter TCA and it is used for glucose synthesis, right? Okay, and acetoacetate is used for further fat synthesis, right? So that's why phenylalanine is known as both glucogenic and ketogenic, because the products can lead to synthesis of both glucose and fats, glucose from fumarate and fat from acetoacetate. So this is the end. So please like and subscribe the channel and don't forget to press the bell button below. Thanks a lot, best wishes and keep on studying. God bless.